So this first slide in this first screencast on recombinant DNA techniques or genetic engineering looks at some of the tools that genetic engineers use to be able to cut out bits of DNA from one organism and place them into another. So the first slide deals with what the term recombinant DNA actually means. Recombinant, sorry, I spelled it incorrectly there. Recombinant DNA is very simply the combination or combining DNA from two different organisms. Okay, so ideally we take DNA out of what we call a donor cell, and we cut that DNA out, and then we insert that into the DNA of our recipient or host cell, and then that recipient or host cell can then express that gene and produce the particular protein that was produced by the donor cell. So the great example that we always used at GCSE was insulin. So if we took insulin out of the gene from insulin out of a human cell, inserted that gene into a bacterial cell, we then can get that bacterium to transcribe and translate the gene and make insulin, which is the way that insulin for diabetics is now made. Okay, so the principles by which this can happen is due to the fact that the genetic code, which we talked about in the previous module, is universal. Well, it isn't actually universal, it's almost universal, but for most organisms it's the same. Okay, and this offers the prospect of genes from one species being expressed in the same way in another species. Okay, so the fact is that an E. coli will read a particular genetic code in exactly the same way as a human cell does, and therefore would the gene for making insulin from a human would be translated or transcribed and translated in exactly the same way in a bacterium, and therefore the bacterium could also make that protein insulin. So essentially what happens is the DNA or gene is taken from one species, and as we'll see that's quite a complicated process, and the DNA or gene is inserted into the cell of another species, and again we'll give you the detail of how we do that, where it combines with the host DNA. And then the recombinant DNA as it now is, because the separated bits of DNA have been recombined in the host cell, can be expressed by that host cell, and we can synthesize a protein. Looking at the next slide, this gives us an overview, and we'll come back and look at a lot of these processes in a little bit more detail in later slides, but it gives us a little overview of how the recombinant DNA, how we actually manage that. The first thing is we have to obtain a gene. We then use a thing called a vector to get that gene into our host cell, and we need to be able to get that vector into the host cell. So dealing with each of these in turn, first of all obtaining the gene. Obviously it's a bit like a needle in the haystack looking for example, the gene for making insulin in the whole human genome. So there are various ways that we could go about doing that. First of all, one of the easier ways of doing it is actually to isolate the messenger RNA. So if we could isolate messenger RNA from a beta islet of Langerhans cell, the messenger RNA for making insulin, okay, we then treat that with an enzyme which is called reverse transcriptase, which does exactly as it suggests. It does the reverse of transcription, i.e. converts the RNA into DNA. We can actually make our DNA. Okay, so that's quite a straightforward way of doing it. Another way, which is quite common these days, is a thing using an automated polynucleotide sequencer, which is a machine which we can actually program the machine to make a piece of DNA with a specific base sequence on it. So as long as we know what the base sequence of our gene is, we can use one of these automated polynucleotide sequencers to make our piece of DNA for us. Thirdly, and a more involved and complicated way, which I'll come back and tell you about in a little bit more detail, is to use a thing called a DNA probe, which in short, is a small piece of DNA of a known sequence which is complementary to the gene we're trying to find, which has a radioactive or fluorescent marker on it, so we can actually track where it is. Having worked out our gene, we need to then get our gene, having isolated our gene, we need to get our gene into the host cell, and we need a thing which we call a vector to do that, which if you know, it's a bit like a sort of Trojan horse, which carries the gene, the target gene, into the host cell. We often use structures called plasmids. Plasmids are naturally occurring loops of DNA that occur in bacterial cells, and what we do is we cut open the plasmid, insert our target gene into the plasmid, okay, using these things called restriction enzymes, which I'll come and talk about later. We can then insert our gene into the vector, plasmid or vector, using another enzyme called DNA ligase, which knits together two bits of DNA, and as well as using plasmids, we can also insert genes into viruses, and we'll come back and look at that in a little bit more detail later on. This slide focuses a little bit on these processes here: electroporation, microinjection, and viral transfer. So I'll deal with each of those in turn. So first of all, before I do that, diagrammatically, here's what I've just talked about up there. We get our DNA, so let's say this is a beta islet of Langerhans cell. We get the DNA out of that cell. We isolate the gene for making insulin, okay, from that. Okay, we then insert that gene into our plasmid. Okay, this plasmid we've got out of a bacterial cell. We've cut that plasmid open. We've inserted the gene into the cut plasmid. We've then taken our gene, which carries our gene for making insulin, and then we insert that into another bacterial cell, and the idea is this bacterial cell can then grow, multiply, and can, if supplied with the correct amino acids, will produce the protein insulin for us. One of the easier ways of isolating the gene was, I said, to start off with the messenger RNA. So if you had our gene in our beta islet of Langerhans cell, that makes mRNA, okay, and that mRNA, if it's isolated, can then be treated with an enzyme called reverse transcriptase, which then makes our DNA. We can then reproduce this DNA using a process called PCR, which I'll come back and explain to you later. And we can make lots and lots of copies of our DNA, and we can then insert those DNAs into our vectors or plasmids, and then insert those plasmids into cells. This diagram here shows us a little bit more detail of how we insert a gene into a plasmid. The answer is we use an enzyme called a restriction enzyme, which cuts our genes on plasmids where it finds specific base sequences. So in this case, wherever it finds this base sequence here, okay, it causes a cut, and it causes what we call a staggered cut. So it leaves a piece of DNA which is single-stranded, okay, and this is called a sticky end. And the advantage of sticky ends is it enables us to stick two bits of DNA from different origins together by complementary base pairing. So this sticky end here is complementary to this sticky end there. Okay, so this could be the DNA of my plasmid, and here's the DNA of my insulin gene, and they've got complementary sticky ends, which means that they can stick together. And the last thing we need to do is we need to stick this together, and an enzyme called DNA ligase completes the sugar phosphate backbones here to stick that properly in. And this shows what we're doing here. So here's our gene for insulin, which we've cut with our restriction enzyme, which cuts it at each end of that. We cut the plasmid open, okay, once here, and that via sticky ends enable us to insert that into that, like that. We'll come back to this later as well. I've got a nice video of that later on, and then we use DNA ligase to knit it all together. The next problem is getting the plasmids into the actual cell itself. Okay, there's a number of processes. This one here is called electroporation. Okay, which word is on the slide that you've got a hard copy of. So you can imagine these green squiggly bits are the plasmids. I then expose my cell, this is the host cell, to an electrical pulse, which causes temporary little pores to form in the cell membrane, which allows our plasmids with our target gene to go into the cell. Okay, and after you switch the electric field off, the membrane then all fuses back together again. So we now have our plasmids inside our cell. The next process is known as microinjection, whereas we get our target cell here, which is going to be our host cell, okay, and we hold it still by suction with a pipette. Okay, so we get a pipette and then suck it onto the end of the pipette, and then we use an injecting device, okay, a microinjection needle, okay, which inserts our plasmids, our DNA, directly into the nucleus of our host cell. Okay, so that's known as microinjection, that process. And lastly, we can use viruses. Okay, viruses work by inserting DNA into cells. That's what they naturally do. So we make use of that by inserting our DNA into a viral DNA, and that virus will infect a cell by inserting its DNA, including our target gene, into the nucleus of the host cell. Okay, lots of gene therapy, which we'll come and look at later, is gone about in this way. Moving on to the next slide. The next slide looks at these things called restriction enzymes, which I've mentioned fleetingly in the last slides, but this looks a little bit more detail about how these restriction enzymes actually work. Restriction enzymes are very useful tools for genetic engineers for a number of reasons. First of all, they are an important tool in enabling us to isolate a particular gene amongst the donor organism's genome. So, going through my example, being able to cut out the gene for insulin in amongst all the other genes that occur in the human genome, we can use these restriction enzymes to do that in ways that I shall illustrate in a second. Okay, so how they work is that restriction enzymes cut at specific base sequences. We call these restriction sites. Okay, and the really useful restriction enzymes cut DNA unevenly. Okay, which causes what we call a staggered cut, and that leaves these little bits of single-stranded DNA sticking out the end of a gene, and we call those sticky ends. Okay, and sticky ends are useful for reasons as it enables us to combine segments from different bits of DNA together. So, for example, sticking our human insulin gene into a piece of plasmid DNA, we were able to do that because of the complementary sticky ends on the plasmid and the insulin gene. Okay, and there are different types of restriction enzymes, and they're different by the fact that they choose different base sequences to cut at. Okay, which can be quite important in other reasons, which we'll come to talk about later when we're talking about how we actually worked out the human genome. But we'll come back to that later. So diagrammatically, here I've got a piece of DNA, and here I've got what we call a restriction site. Okay, and what we often find is that a restriction site for these restriction enzymes which produce these staggered cuts often recognise sequences of which are what we call palindromic. Okay, so we've got here is the opposite of here. Okay, so that is so GAATTC and GAATTC. Okay, so that will cut where at that particular site, and so it's cut here and here, which has created this staggered cut here, and which creates these sticky ends, which are single-stranded bits of DNA that stick out the end here. Okay, so that's what restriction enzyme does. Moving on to the next slide. The next slide looks at a process called electrophoresis, which is the way that we can separate fragments of DNA. So if we've treated, let's say, the whole human genome with our restriction enzyme, what we're going to end up with is a whole series of fragments of DNA which are all different sizes. Okay, and the next thing that we need to do is to be able to separate all those different bits of DNA, and we do that by separating them by size by a process called electrophoresis. Okay, and that involves placing our DNA fragments into a little well, or a little depression in a piece of gel, gel made of a substance called agarose. Okay, so we cut a little well into a piece of agarose gel. Okay, and then we put our gel in a tank containing a buffer solution, and then we place a voltage across the gel, and because the DNA is slightly negative, the DNA will migrate through the gel towards the positive electrode. Okay, and what we find is the shorter DNA fragments migrate faster through the agarose gel than the larger ones. So what happens is that we separate our DNA by size. Okay, and if we actually stain the bits of DNA, we can actually see where the DNA are. Okay, we can also then adsorb the DNA onto a nylon sheet, which is a process known as southern blotting, and I'll come back and explain that a little bit later. So, illustrating all of that in a diagrammatic form, hopefully this illustrates what I've just said. Here we've got our DNA sample. We treat that with our restriction enzymes, which cuts it up into different size fragments. Okay, we then put all these fragments into this little well here. Okay, the well here has been cut into this block of jelly-like material, which is made of a substance called agarose. We then pour in a buffer solution. Okay, and then we put a voltage across this. Okay, and what happens is the DNA will then start to migrate through the gel towards the positive electrode, and the smaller bits of DNA will manage to go further than the larger bits. Okay, so our DNA will be deposited at different lengths along this gel. Okay, so just going through the diagram here, so the DNA segments are loaded into wells in a porous gel. The gel floats in a buffer solution within a chamber between two electrodes. When electric current is passed through the chamber, DNA fragments move towards the positively charged electrode. So here we have, let's say here, we've got our different fragments of DNA moving along here. So this bit here is smaller than this bit here because it's managed to travel further through the gel. Okay, as it says here, smaller DNA segments move faster than the larger DNA segments. And I've got a little video clip which illustrates some of that as well, which I'll show you. So this bit, I've put my gel in here. I put my gel in here and allowed it to solidify. Okay, so I've heated up my agarose gel so it's warm, so it's liquid. I then put it into this bath here. Okay, and then what I do is I put this comb structure into my liquid gel or liquid here. I then let the liquid solidify, and then I carefully remove the comb, leaving these wells in our gel, which we can then put the DNA into. So here's my prepared gel here. This is now solidified agarose gel with these little depressions called wells at one end. And I can put, let's say, DNA into this well here. So here I've got DNA from probably different sources, and I'm loading the DNA into each of these wells here, and there's a dye in here which shows us, which will pass through the gel and show us when the gel is finished, which will show you that in a second. So here I've got a gel, so different bits of DNA here have been transferred or go at different distances through our gel here. On the left here I've just got a scale, so these are all bits of DNA of known length, which provides a little scale so I know that this piece of DNA is roughly the same size as this piece of DNA here, and I actually know what the size of this piece of DNA is. So we get this pattern. Okay, so what we've done is separated the bits of the DNA, which creates a pattern which can be very useful, as we'll discover in a second. Okay, so the last slide on this particular screencast looks at the idea of how we can identify segments of DNA. So what we're trying to do here is we're trying to look for a specific gene. So using our example of insulin, let's say we've chopped up our human genome into all sorts of different fragments, and we've separated all those fragments on our by electrophoresis. Now we want to actually find which of those fragments is the gene for insulin, and we can do this by using a thing called a DNA probe. Okay, and a DNA probe is a single-stranded segment of DNA which is complementary to our gene, the gene that we're looking for. So we need to know at least a section of our gene, of what the gene sequence is, or the sequence of nucleotides on our gene that we're trying to find. We actually need to know that. We then create a short segment of single-stranded DNA which is complementary to our gene, and then we put a fluorescent marker or a radioactive marker onto that probe, okay, so we can know where the probe is. Okay, now because of our probe is complementary in sequence to a portion of our gene, okay, the probe, if it's mixed with all the DNA, will complementarily bind, or by complementary base pairing will bind or anneal to our the gene that we're trying to find. Okay, so this enables us to locate genes, okay, amongst all the fragments which we've separated by electrophoresis. Okay, that sounds very complicated. I'll try and explain that by using diagrams. Okay, so here's quite a simple diagram where what I've done is I've chopped up all my genes, okay, creating all these different fragments. I've separated all these fragments, okay. I then have to make those fragments single-stranded, which I can do by immersing these in weak hydroxide solution. And then what I can do is I introduce my probe and wash it over my fragments, and it will bind specifically to the gene that I'm looking for. Okay, so this probe has specifically bound here, here, and here, which tells me that those are positions where I would find my gene, for this case, insulin. Okay, so I know these, and these are genes for insulin. Okay, just going into that little bit more detail of how you actually do that. Okay, so the gene fragments which we've created by using restriction enzymes are separated by electrophoresis. Okay, the electrophoresis is then, so the pattern on the electrophoresis is transferred to a nylon sheet by a process known as southern blotting. Okay, and the DNA on your nylon sheet can then be converted to single-stranded DNA by treating with hydroxide ions. You then add your probe. Okay, so you pour your probe, you would immerse your sheet into a solution containing the probe, which is a short length of single-stranded radioactive or fluorescent DNA known to be complementary to the sought-after gene. Okay, so you add that. The probe will then anneal, which means bind to or hybridize, same word or same meaning, to the sought-after gene by complementary base pairing. What you then do is to wash off any excess probe off the sheet. Okay, so you've only got the probe on your sheet which is actually bound to the gene you're looking for, and the nylon sheet is then exposed to a photographic plate which reveals the position of the gene on the sheet, and then the position of the gene can be traced back to the electrophoretic gel, so we know the position of our target gene on our electrophoretic gel. Okay, and then that can be extracted from the gel. So diagrammatically, what we've done there, so we've got our, sorry about that, we've got our DNA which we've cut up with restriction enzymes. We then separated those. Okay, we separated those by electrophoresis. We've then placed a nylon sheet on top of it. Okay, and the DNA blots up onto the nylon sheet and attaches quite strongly to the nylon sheet. It's called southern blotting. Okay, we then treat the sheet with weak hydroxide to convert all the DNA into single-stranded DNA. We then add our probe, okay, to our sheet, and the probe will bind to wherever it comes across complementary bits of DNA. We then wash off any excess probe and put a photographic plate on top of our nylon sheet, okay, which then shows up where the probe actually is. Okay, so from that we know that the probe has stuck to the DNA here and here. Okay, and from that we know that the DNA that we're interested in, okay, is the DNA here and here. So we know that our gene for, let's say in this case, insulin, can be found here and here. Okay, and we can then remove that DNA from the electrophoretic gel, okay, with a syringe, okay, and we can then treat that with our restriction enzymes, okay, and stick it into a plasmid, and that can be used to transform the host cell, the bacterial cell.